everyone, this is Dr. Karen's, and today I'll be discussing Chapter 13 for Biology 2420, Microbiology for Non-Science Majors at Eastfield. So this chapter is discussing viruses and a few other things, which we'll get to at the end. Viruses are included in microbiology because of really how small they are and the fact that they cause disease. But remember that unlike the other types of microbes out there, bacteria, protozoa, fungi, things like that, viruses are not technically alive. They're not cells. They do contain either DNA or RNA and some protein, but that's about it. They're a lot smaller than bacteria. They do not have very many genes encoded in their nucleic acid, and so they cannot do any of the processes that a cell would be able to do and what makes a cell alive. So they can't reproduce on their own. They can't metabolize on their own. They can't respond to the environment. So that is why they're not considered to be alive. However, they do cause disease, and so they're important when we're studying microbiology. So let's look at some terms. So I tend to use the term virus all the time when I'm talking about viruses, but if you really want to be technical, there are two different states for a virus. There's the extracellular state, and then there's the intracellular state. So since viruses are wanting to invade a host cell so it can use the cell's machinery to make copies of the virus, you have to kind of consider that virus in that intracellular state as well. So the extracellular state of a virus is technically called a virion, but let me just say that often I'll use that term virus. But the technical term is virion. It consists of the nucleic acid, and depending on the virus, that nucleic acid can be DNA or it can be RNA. And then that nucleic acid is surrounded by a protein coat called a capsid. Some viruses, particularly animal viruses, might also have an additional membrane around that capsid called the envelope. And so we'll look at that in a second. When that virus has entered a host cell, it exists only as the nucleic acid, and it's then that virus can take over the cell, direct the cell to kind of forget what the cell is supposed to be doing and instead focus all of its energy and resources on making more viral copies. That is really the goal of a virus, is make more copies of itself. It can't do it on its own, so it needs a host cell to do it. If that virus is made of DNA, in order to make more copies of that DNA replication has to occur. The virus can't do that, but a cell knows how to do that and has the enzymes required for that. Part of making more virus means making more of that capsid. That's protein, so that means transcription and translation needs to happen. The virus can't do that, but a cell knows how to do that. And so the virus, once it's in that cell, it'll take over and direct that cell to only focus on DNA replication, transcription, and translation of the virus. Here's an example of a virus and its capsid. So in this case, this looks like a double-stranded DNA virus surrounded by this particular shape of the capsid. A capsid can take on different shapes. It just depends on the virus. So I mentioned before, viruses can be made of DNA, but they can also be made of RNA. Never both. It's one or the other. And depending on that virus, if it's a DNA virus, it could be double-stranded DNA. It could be single-stranded DNA. It could be double-stranded RNA, which sounds weird because normally we don't see that, but in a virus it can exist. Or it could be single-stranded RNA. So a lot of different varieties for that viral genome. That nucleic acid can be circular, can be linear. It's just a wide variety. this is showing, we've seen this picture before, but here's an E. coli cell. Here is the part of the genome from the E. coli cell, so part of the DNA. And here is showing you how a viral genome would compare in size. So we're really talking about a really small amount of nucleic acid, and honestly only enough genes to direct, to code for the proteins that you need, that the virus will need for the capsid, is basically it. And maybe attachment proteins too. All right, so what kinds of cells will viruses infect? There are viruses that can infect bacteria cells. We've learned a little bit about them already. They're called bacteriophages. There are viruses that affect plants, viruses that can infect humans, viruses that can infect other animals. Normally, there's just one host for a virus, so they're very specific in terms of which organism cells they can infect, and then even within the organism. So if you think of human viruses, so let's think of like the flu versus even the cold. The flu virus infects lower respiratory cells. Viruses that cause the cold will infect upper respiratory cells only, and they don't switch. It's very specific usually. There might be some viruses that are more general and can infect different types of hosts, but usually it's pretty specific. This is figure 13.4. It's giving you, again, a size comparison. So they're showing you this red blood cell. Compare that to E. coli. And then viruses are even smaller than that. And in the viruses shown in this image, they all look very different. They're even different sizes from one another. And that has to do with the capsid. What is the arrangement of the proteins surrounding those nucleic acids? All right, so going back to those capsids. So this is protection for the viral nucleic acid. This is going to be how the virus is going to be able to attach to a host cell. So there'll be proteins embedded within that capsid that can recognize different receptors on the host cell. The protein subunits that make up a capsid are called capsimeres. Could be made of one type of protein, many types of proteins. It just depends on the virus. And that shape or the morphology of the capsid can be a variety of different shapes. They don't have a picture here. Oh, I guess they do have a few pictures, but sometimes that capsid is helical, polyhedral, so like a 3D shape with sides to it, complex. So you can see here, this would be helical. Up here, it's hard to see the helix because it's kind of zoomed out. This is figure 13.5. Bullet shaped, this would be like a complex shape. It just depends on the virus, what shape or morphology that capsid is going to take. This is a very interesting virus, a very interesting capsid. This is bacteriophage T4. So this is a bacterial virus. It looks like an insect, and it kind of works a little bit like an insect too, but a very complex capsid. The viral genomes, this will be a DNA virus you'll find inside the head of this, but there's a tail with tail fibers, and you can kind of see that's how the virus is going to attach to the host cell. This is very specific for this particular bacteriophage. All right, some viruses, animal viruses more so than bacterioviruses. So a lot of animal viruses will also have an envelope that is basically cell membrane that came from the host cell, and you'll see how in a little bit. But that envelope is basically made up of the components of the cell membrane of the host cell, and it's outside the capsid, so it's another layer. It will have some proteins embedded within that envelope that are encoded by the viral genome. Those are called glycoproteins. And the glycoproteins are going to help with attachment to the host. So not all animal viruses have an envelope, but those that do are going to be a little more fragile because of that membrane. So here's an example of an enveloped virion. So What's inside is a helical-shaped capsid. The nucleic acid is inside of that. And then this big round thing is the envelope. And look, it's made up of a phospholipid bilayer, just like a cell membrane is. But we have these spiky glycoproteins sticking out of it, and that's going to help with attachment. This can also serve as, if you're thinking about developing a drug against an animal virus, if it has an envelope and it has these glycoproteins, these are viral proteins, so it could be a possible target for a drug. All right, this is table 13.1, just gives you a list of different characteristics and properties of a virus and then compares that to a cell. So this is a good review just to see what is the difference really between a virus and a cell. How do we classify viruses? So if you recall from some of the earlier chapters, there are some pretty extensive classifications of bacteria and even of other organisms as well that we don't talk about in this class. But for viruses, there's not a set defined way of classifying them. Usually, at least in our book, they've chosen to use a classification based on the type of nucleic acid they have, and then they might have to go further, like, do they have an envelope? What is their shape? What is their size? But it's not as extensive or as detailed of a classification system as you see among other organisms. All right. So the purpose, like I mentioned before, the purpose of the virus's goal is to make more copies of itself. And just keep in mind, what does that mean? That means the virus is made up of viral nucleic acid and a protein capsid. So that means when the virus is wanting to make more copies of itself, it means that more viral nucleic acid or viral genome has to be made, and more viral protein has to be made. And those processes that do that, DNA replication if you're a DNA virus, transcription and translation if you are trying to make protein, those are all processes that the cell or the host cell already knows how to do, but the virus can't. But that's what has to happen inside the host cell. So the virus is trying to get into the host cell, take over that host cell, so more copies of that virus and virion can be made, and then it wants to leave the host cell so it can go and infect more host cells and make more copies and so on and so forth. And so this replication cycle kind of describes the things that have to happen in order for all those things to occur. There are two basic types of viral replication processes, and I need to say that the way this book is organized, it talks about these processes with respect to bacterial viruses or bacteriophages, okay? So even an animal virus or a plant virus still has to undergo the same basic steps of the replication process that we're going to talk about, but just keep in mind that the specific examples they're showing you in this book are bacteriophages, and that means a DNA virus, okay? So one of these replication processes is called the lytic replication cycle. So lytic means lyse. So that means that once the virus enters the host cell, makes more copies of itself, and leaves, it causes lysis of the host cell, and that means the host cell is going to die. So I know that we're talking about bacteriophages right now, but just think about what that means for that cell and that population of cells. Whatever that cell is designed to do is not going to be able to do it anymore because the virus takes it over and then kills the cell. So what are the five main things that have to happen? First, that virus has to attach to the host cell. The viral genome, that DNA in this case, but if we're talking about an animal virus, then it could be RNA. The viral genome has to enter the host cell. And then synthesis has to happen. Synthesis is the making of more virus. So making more viral genome, making more viral proteins. And then assembling those things into virions. and then those will be released. So let's look at the picture here. So they're using an example. This is figure 13.8. This is basically what the lytic replication cycle looks like, using as an example T4 bacteriophage. So in the attachment step, this particular bacteriophage it looks like a little bug. It kind of attaches to the host cell, and almost like a syringe, it then will inject its genome, in this case DNA, into the host cell. That's the entry step. Once that entry of the viral genome occurs, notice how the bacterial chromosome is directed to be destroyed. That is coming from signals from the viral DNA. So the bacterial chromosome is destroyed, and now that means that cell is no longer doing the work that the bacterial chromosome was telling it to do. Instead, it's the viral DNA or the phage DNA that is now giving the orders. And those orders are, make more copies of me. So the synthesis stage, we need to make more copies of viral DNA, and that's going to be done using DNA replication. That's how you make DNA. And then we need to make more viral proteins to put together this capsid. Proteins are made with transcription and translation. And so the viral DNA is going to tell that cell, transcribe and translate me so I can make more proteins. Then all of those things have to assemble themselves into virions. And then those virions are getting released. Now there's a lot of them in that cell. Instead of just one, they burst from the cell lyses and dies. Figure 13.9 is showing you, this is just a graph looking at how much the number of virions will increase during this cycle. So if you start with one entering the cell, by the time they get released, you can have, what is this, like 500? A lot. Start with one infecting that cell leads to 500 or more leaving that cell that can go on, and each will find another host cell to infect. Okay, so that was the lytic cycle. Some bacteriophages will, they still have to go through the same basic 5 steps, but there's a little bit of a side step involved, and it's more like a dormant phase of this cycle. And so this is called the lysogenic cycle. So it's similar, it starts off and ends the same as the lytic cycle, but there are some additional things that happen in the middle. And really the difference is the virus kind of becomes dormant and quiet for a bit before it wreaks its havoc on the cell. So that dormant, inactive form of the phage is called a prophage. So let's look at this picture. The example they're using here is the lambda bacteriophage. So this one in particular will replicate using the lysogenic cycle. So it starts with attachment as usual, entry of the phage DNA. This time that entry does not tell the host cell's DNA to disintegrate. Instead, it's almost as if that host cell has no idea it's been infected. And so after this entry phase or stage, this is when the virus goes into lysogeny, or this dormant state, and it becomes a prophage. So the viral DNA has entered the cell, but instead of taking over the cell, it just incorporates itself into the host cell's chromosome through homologous recombination. And so the cell is not aware it's even affected or infected. And instead, the cell goes on and does what it's supposed to do. It'll divide and divide and divide and divide and divide. And remember, before cell division can take place, DNA replication has to occur. And when that happens, since that viral DNA is now part of the host cell's chromosome, that gets replicated too. So every time the bacteria divides, it creates another copy of that viral DNA. And so at some point, some signal kind of tells that virus that, hey, it's time to kind of pick up where we left off in this cycle. And so that's called induction. And that there's some signal, it could be stress, environmental stress on the cell, environmental changes, so conditions are changing. It could be a variety of things that tells that viral DNA to jump out of the bacterial chromosome and start taking over the cell. And now the bacterial chromosome is destroyed, and then we pick up where we left off in that lytic cycle. So now we have synthesis of the viral DNA and viral proteins, assembly into virions, and then release. And that does end up killing the cell. So why would some viruses be able to do this lysogeny? I think it just depends on what genes are in that viral DNA. How long does lysogeny occur? Again, I think it depends on the virus, and it probably depends on the environmental conditions, nutrition conditions, things like that of the cell. But the end, eventually, if you're going to make more copies of the virus, they have to leave that cell somehow, and it's going to end up killing the host cell. All right, so those are two different replication cycles, the same basic 5 steps. We were talking about bacterial viruses in those cases. Now we're going to shift over to animal viruses. So animal viruses, they still need to do the basic 5 steps. They have to attach to a host cell. They have to enter, the genome has to enter the host cell. Synthesis has to happen. Assembly has to happen into virions, and then those virions need to be released by the cell. However, animal viruses can vary more than bacterial viruses, and so this process can be a little more complex. If an animal virus has an envelope, then entry can happen in a few different ways, and I'll show you in a second. Likewise, if a virus has an envelope, exit from the cell can happen in a variety of ways. And then also, because animal viruses can be made up of DNA or RNA, and the DNA could be double-stranded or it could be single-stranded, same with the RNA, there's a lot of different possibilities for the identity of the viral genome, the makeup of that viral genome. And so that's going to make that synthesis step vary among viruses as well. And that's a little complicated, and I'll try to talk you through that in a second. But first, let's talk about the entry of an animal virus. So attachment has to happen no matter what. So that step is always going to happen no matter what type of a virus you are. But if you're an animal virus, entry of that viral genome can happen in a variety of ways. So if you are an animal virus without an envelope, then the entry of the viral genome happens via something called direct penetration, which is basically just how it happens with the bacteriophage. The capsid attaches to the host cell, and the viral genome is kind of injected into that host cell, and the capsid remains outside. That we saw that with the bacteriophages. But if you're an enveloped virus, things can be different during entry. So in membrane fusion, this is one form of entry where we have our virion that's attaching to the host cell, but it's the envelope, which is made up of cell membrane components. That is what's attaching to the viral or the host cell. In membrane fusion, that envelope will fuse with the host cell membrane and kind of open up, almost like, well, not quite like endocytosis, but it'll open up and allow the release of both the viral genome and the capsid into the host cell. So now we have the capsid coming with that viral genome into the host cell, which we haven't seen before. There's an extra step now required in order for the virus to take over the cell, because it's only like naked viral genome that can take over a host cell. So in order for that to happen, the virus needs to uncoat, or that capsid has to come off and uncoat from the viral genome. That's what that uncoating step is. If you recall from chapter 10, we were talking about different antiviral medications. Some of them will target this uncoating step. So if a drug prevents this from happening, then even though the virus has entered the cell, it cannot take over the cell and therefore will not be able to make more copies. So you can kind of stop an infection from spreading that way. So that's one way an enveloped virus can enter a cell. Another way is through true endocytosis. So the envelope of the virus attaches to the host cell, and the cell membrane completely engulfs all three things: the envelope, the capsid, and the viral genome. All three of those things enter the cell. So you have to add even one more step, even in addition to uncoating. First, you have to have fusion between the cell membrane that formed the vesicle, the endocytic vesicle around everything, fusion of that and the envelope, so that can open up and release the capsid and the viral genome. And then you have to have uncoating of that viral genome in order for the virus to take over. So again, because animal viruses may be enveloped, it provides additional ways of entry into a host cell. For the same reasons, I want to skip ahead to the release of virions, of animal virus or animal virions from a host cell. The same reasons that complicate entry can complicate exit. I'm going to skip around a little bit. If you are an enveloped animal virus, how do you acquire that envelope? You acquire it upon exit from the host cell. So the virus has already made more copies of itself and assembled itself in nucleic acid and capsid, and then that capsid and the nucleic acid inside of it will find its way to the host cell membrane, where there are viral proteins now embedded within that membrane, and it leaves via kind of like, we call it budding. It's kind of like exocytosis. So it'll leave and take some of that cell membrane with it, and that cell membrane becomes the envelope with those viral glycoproteins embedded in it. The nice thing maybe sort of about this method is that the host cell doesn't die. The viruses just bud off. And so again, having an envelope can give you different ways of exiting a host cell. If you don't have an envelope, if you're an animal virus and you don't have an envelope, it'll probably lead to lysis when those get released, like we saw with the bacteriophages. Okay, so animal viruses can enter in different ways, and they can be released in different ways. They can also do synthesis in different ways. So now I'm going to go back and just remind you that, but let's think back to the bacteriophages. So those were DNA viruses, double-stranded DNA viruses. So when that double-stranded DNA takes over the host cell, two things need to happen in order to make more copies of the virions. You need to make more viral DNA, and you need to make more viral proteins. That is very straightforward to do because the virus is made of double-stranded DNA, which is very familiar to a host cell, right? Especially an animal cell. It's made of double-stranded DNA, or the chromosomes are double-stranded DNA. It's true for a bacterial cell too. So the cell can very easily make more viral DNA simply by doing DNA replication, right? And the cell can make more viral proteins by doing transcription and translation using the genetic information from the virus. Okay, so that's very straightforward, and those things would already happen normally in a cell, and so it's easy. But animal viruses aren't always double-stranded DNA. But the same thing still has to happen. More copies of that viral genome need to be made, and more viral protein needs to be made in order to make more virions. So what's complicated now is, how do we make more viral nucleic acid? And in order to answer that question, we have to identify, well, what is going to be the template to make more nucleic acid of whatever that form is in the virus? And also, if we're trying to make more viral protein, we need messenger RNA to do that. That's where transcription begins. How do we make more messenger RNA so we can make more viral proteins? These questions, the answers to these questions will vary depending on what type of virus you are. And what I mean by virus, I mean what type of nucleic acid you're made of. So we're looking at figure 13.13. It is showing you the synthesis step of viral replication for 3 different types of animal viruses. These are all RNA viruses. So they're not showing you how it works for a double-stranded DNA virus, but let's talk through that first. Let's say that we had a double-stranded DNA animal virus. So the double-stranded DNA enters the host cell. In order to make more of that double-stranded DNA, that process is DNA replication in order to do that. So the template used to make more of that double-stranded DNA is going to be the 2 strands of that double-stranded DNA from the virus. That's the template that's used during DNA replication. You start with 1 double-stranded DNA molecule and you get 2 out of it. The template are both strands of that original DNA molecule. Now, if you're starting with double-stranded DNA and you want to make protein, well, how do you do that? Well, you have to make messenger RNA first, right? Using one of the strands of the double-stranded DNA. So you do that regular transcription, just like you learned in chapter 7, and then move on and do translation. That is straightforward. What figure 13.13 is showing you is how it's not as straightforward when the virus is something other than double-stranded DNA. So let me walk you through this. So we're looking at part A. In this example, the virus is positive single-stranded RNA. What does positive and negative mean? Positive and negative means complementary. So if you go back to our double-stranded DNA molecule, our example, a double-stranded DNA, those 2 strands are complementary to one another, right? It doesn't matter what kind of DNA you're talking about, where it's from. If it's double-stranded DNA, each of the strands is complementary to the other, which means they can base pair with each other. So one of those strands is positive, one of those strands is negative, okay? That can be true for RNA as well. The positive single-stranded RNA is always going to act as messenger RNA. So even if you are just a regular cell, you're made of double-stranded DNA, and you want to do transcription so you can make proteins, you are going to make messenger RNA to do that, and that messenger RNA is going to be positive single-stranded RNA, okay? So just take that as fact. Positive single-stranded RNA will be used directly as messenger RNA. All right, let's go back to this example. So this positive single-stranded RNA virus has entered the cell. More copies of this positive single-stranded RNA need to be made because that's the viral genome. We had to make more copies of that. And we also need to make viral proteins. All right, let's start with the viral genome. How do we make more positive single-stranded RNA? Remember, anytime you want to make any type of nucleic acid, you have to have a template. And the template has to be complementary to what you are trying to make. So if we're trying to make more positive single-stranded RNA, the cell has to be told, you need to make negative single-stranded RNA first, because that's complementary to what you're trying to make. This negative single-stranded RNA is going to serve as the template to make more positive single-stranded RNA. Okay? The other thing we want to know is what is the cell going to use as messenger RNA to make more viral proteins? Well, what I told you before is messenger RNA is positive single-stranded RNA. Since that's what has entered the cell in the beginning, this can be used directly as messenger RNA. Transcription can occur immediately on this piece of, translation can occur immediately on this piece of viral genome to make viral proteins. Now let's look at another example. This is part B. In this example, the animal virus is made of single-stranded RNA, but it's negative. But the same thing still has to happen. The virus wants to make more copies of itself, which means in this case, it wants to make more copies of this negative single-stranded RNA, and it wants to make more viral proteins. Okay, so how do we do that? How is the cell going to do that? So in order to make more copies of negative single-stranded RNA, the cell has to be told to make the complementary strand of RNA first, which is going to be positive. It's complementary to that negative strand, and this is going to be the template to make more of that negative strand. This positive strand that was made will also serve as the messenger RNA during translation to make more viral proteins. And then in the last example here, the animal virus is made of double-stranded RNA. All right, we need to make more double-stranded RNA. Anytime you have a double-stranded nucleic acid, whether it's DNA or RNA, both strands need to come apart and both strands need to serve as templates so you can make more copies of both strands. And that's what will happen. The two strands will unwind. One is positive, one is negative. Both of them are used as templates to create more of the double-stranded RNA that we started with. And because one of them is positive, single-stranded RNA, that's the strand that will be used as messenger RNA during translation to make more viral proteins. So this is really, it's a bit complicated, but the point is to show you that no matter what type of nucleic acid the virus is made of, in order to make more copies of that virion, you need to make more copies of the viral genome, which is the nucleic acid, and you need to make viral proteins. The question is, how complicated is that if the viral genome is something other than double-stranded DNA, which the cell already knows how to deal with? So if you've got more questions about that, I suggest reread the book. You can definitely send me an email and see if I can try to explain that better to you. You could also look on Google, look online, look on YouTube, see if there are some animated images, and that might make a little bit more sense to you as well. Overall, though, an animal virus, it still wants to do what a bacteriophage wants to do. It wants to attach to a host cell. It wants to get inside the host cell. It's trying to make more copies of itself. It needs the host cell's machinery to do that. And in order to make more copies of any type of virus, remember, a virus is made of a nucleic acid and proteins, so you need to make more of both. Put those things together into virions, and then they need to be released from the host cell so they can go and infect other cells. Let me talk about one other thing here before we move on. It's something I almost forgot about. Some animal viruses are what we call retroviruses, which are really interesting. So a retrovirus is a type of a positive single-stranded RNA virus. So it looks like this, part A of figure 13.13. But instead of just that positive single-stranded RNA being used directly as messenger RNA to make proteins, it'll do transcription in reverse, meaning it'll use that the cell will take this positive single-stranded RNA from the virus and turn it into double-stranded DNA first, and then it will do transcription to make more positive single-stranded RNA and then translation. Why would it do that extra step? It's an advantage to the virus, especially if you're talking about a virus that infects a host cell that contains double-stranded DNA in their chromosome, and all cells do. This case, if you have a virus that exists as single-stranded RNA, but it can turn itself into double-stranded DNA, now that virus has the ability to incorporate itself into the host cell's chromosomes and become dormant, kind of like what we saw in the lysogenic cycle for those bacteriophages. HIV operates like this, which is why the incubation period is so long. It could be 30 years before you start experiencing symptoms from HIV. And that's because it's an RNA virus, but it can turn into double-stranded DNA and insert itself into your chromosomes. It's very advantageous for a virus to be able to do this. It's very disadvantageous to us. So going back to that concept of dormancy, so if you're talking about the ability of an animal virus to become dormant, we call that latency, not lysogeny like we saw in the bacteriophages. So what is different about this? Same thing will happen in the sense that the viral double-stranded DNA incorporates itself into the host cell's chromosomes. But if you're talking about an animal virus, that incorporation is permanent. And that's what latency is. The incorporation into the host cell's chromosome becomes permanent, and there could be some immune stressors that kind of activate the virus, but it doesn't come back out of the host cell's chromosome like you saw in the lysogenic cycle with bacteriophages. So it can be a problem if the particular virus is causing a disease. All right, table 13.4 does a nice comparison of all of these kind of steps and things related to viral replication, comparing bacteriophages to animal viruses. So it's a nice summary where I don't think they make it very clear in the textbook. It's nice to see it in a table like this, and you can look at the differences kind of all on one page. All right, something else that's interesting. Some viruses can lead to cancer. So not necessarily just an infection, but could lead to cancer. So remember, cancer is uncontrolled cell division, and that uncontrolled cell division can spread to more than one cell, more than one tissue. So just to give you kind of a background first, we've got, let's talk about our own cells. We have, in our DNA, we have things called proto-oncogenes. They are promoting cell growth and cell division, and they're going to kind of dictate that cell cycle of mitosis and meiosis in our cells. But if something goes wrong and those proto-oncogenes or the oncogenes get activated and they're uncontrolled, that can lead to cancer. There are chemicals that can cause this, but some viruses can cause this too. So how does that happen? So here's what some DNA in a normal state. There's a proto-oncogene that is there to regulate cell division and cell growth. There is also a gene for a repressor for that proto-oncogene, and the repressor gets expressed, transcribed, and translated, and the repressor's job is to keep the proto-oncogene from getting out of control. And so no cancer because we have that repressor. That's a normal state. What happens when you've got a DNA virus that can insert itself into your DNA? You have no control over where that virus is going to insert itself. So if it inserts itself right near the promoter, or inserts a promoter, it encodes a promoter for this proto-oncogene, now we've kind of activated this proto-oncogene to become kind of out of control. But we're still making a repressor, so we can still kind of turn it off even though it's been turned on, and we don't have cancer. The issue is if a virus inserts itself again, and if it happens to insert itself in the middle of this repressor gene, now we're not making repressor, so now we cannot block this activated proto-oncogene, and now we get cancer. So there are some cancers that are caused by viruses, and it happens in this way. Cervical cancer is I don't want to say a popular one, but it's popular in the sense that it's in the news, and you can see it on TV a lot, because the virus that causes and can lead to cervical cancer, not all the time, but it can, is that human papillomavirus, or HPV. And there's a vaccine against that virus now. So the virus itself can cause an STD, but it could also lead to cervical cancer. And so that's why there's a push for the vaccine, not so much to prevent the genital warts that it causes, but to prevent the possibility of developing cervical cancer. Other cancers that are caused by viruses: Burkitt's lymphoma, Hodgkin's disease, and Kaposi sarcoma, that's caused by HIV. So, interestingly enough, because we know that some viruses can do that, can insert themselves into our chromosomes, we can engineer viruses to carry genes that maybe we're missing. So there are a lot of genetic diseases out there that are caused by genetic mutations that we've discovered, okay, what gene is affected, what exactly is the mutation. We can engineer viruses to carry a normal copy of that gene. Instead of carrying viral DNA, they carry the normal copy of your gene that you don't have, and we can infect our cells with that virus, and it delivers the normal copy of the gene for us. And there have been some success cases with curing some diseases in that way. The very early stages of this technology, though, often what would happen is what we see in figure 13.16. We didn't have any control over where that virus was delivering the DNA, and sometimes that DNA, while it was carrying something that we needed, it would insert itself into another gene that we also needed, and then it would cause cancer. But now we have more sophisticated methods of doing that kind of thing. All right, we're going to skip over culturing viruses. We're not doing that in the lab. It's a lot more complicated because you need host cells. We're not going to worry about that. What I do want to finish with are these other types of microbes that they're not viruses necessarily, but they kind of just throw them in this chapter. And that is viroids and prions. So viroids are like a virus. They're like an RNA virus, but they don't have a capsid. It's just single-stranded RNA. It is infectious, but specifically to plants. And so what happens is when that RNA enters a host cell in the plant, it doesn't code for any protein, so it doesn't do like translation or anything, but that piece of RNA will be able to find complementary pieces of RNA in the plant and bind to them, and then that can lead to reactions from the cell that will lead to disease. So some examples, This is what can happen to potatoes. I'm sorry, I don't really know the name of the viroid in this example. I think tobacco mosaic virus is like that too. And the other thing are prions. These are, if you remember from, it was either chapter 9, I think it was chapter 9, when there was, they gave you a list of different types of microbes in the order of how resistant they are to different methods of control. Prions were at the top of the list. They were the most resistant to any type of treatment. This is because proteins are basically just infectious, prions are just infectious proteins. There's no DNA that encodes them or RNA that encodes them. It's basically kind of what happens is existing normal proteins kind of magically turn into infectious proteins. And there's not really a mutation that leads to that. And that's what makes it difficult to kind of figure out how it happens and therefore how to treat it. So one of the examples they give you of a prion is what's called prion PRP. So the normal version or the cellular version of PRP protein is a protein made by all mammals. I think you find it in the brain. And you need it to function, to do whatever it needs to do. The normal shape of the protein is an alpha helix. That's the 3D shape. Prion PRP is the bad version of cellular PRP. It takes on a different shape in the form of what we call a beta pleated sheet. And in this shape, it can cause disease, most notably different types of brain encephalopathies. So it causes kind of big holes in the brain. What is kind of scary about this prion is that how it originates. And I'm not really sure if scientists really know what triggers this change. But basically what happens is cellular PRP changes into prion PRP. It just refolds itself. Again, there's no genetic DNA level mutation telling that protein to refold. It just, the existing protein, for whatever reason, just starts to fold differently. And the example, when they talk about this in the book, they give the example kind of unfairly of like one bad teenager turns all of the other teenagers around it into bad teenagers. But that's kind of what happens is if one cellular PRP protein changes into prion PRP protein, it causes all the neighboring ones to change too. And so the problem is that not only are we missing the normal PRP, but now we have a different protein that's causing problems. And these are some of the encephalopathies it can cause. So if this is normal brain tissue, prion PRP basically puts holes in your brain. And it's incurable, degenerative brain disease that you can die from. So how do you even get it? So let me back up. Mad cow disease is one of these. It's caused by a prion. If you ingest something that has got prion PRP in it, it'll turn your regular PRPs into prion PRP. Or if you get a transplant with it or something like that. So I guess what I said before wasn't correct. It wasn't that magically normal PRP turns into prion PRP actively in your brain. You have to be contaminated with it first, and then it changes your neighboring PRPs into the prion PRPs. There is no treatment for these diseases for multiple reasons. One, prions are very resistant to a lot of different methods, but also these particular ones are in your brain, and so it makes that even harder when you're trying to treat a brain disease. So just an interesting thing, they're in this chapter because even though they're not viruses, they are very small things that can cause disease. In terms of the study guide, and so we're going to end there. In terms of the study guide, most of this chapter, we're going to focus on the virus stuff. So again, look to the study guide, see what you need to know, And that's it for now, and then I'll see you for chapter 14.